Neural Communication: The Structure and Function of a Single Synapse by Chrissy Wilhelm. A synapse is the junction between two neurons where communication occurs, allowing the transmission of signals throughout the nervous system. This process is essential for coordinating sensory input, motor response, cognition, and various physiological functions. Synapses enable neurons to pass information either electrically or chemically, ensuring efficient communication between different parts of the body and the brain. There are two main types of synapses: chemical synapses and electrical synapses. Chemical synapses, which are the focus of this presentation, rely on neurotransmitter release to transmit signals between neurons. When the action potential reaches the presynaptic terminal, neurotransmitters are released into the synaptic cleft, where they bind to receptors on the postsynaptic neuron, leading to either excitation or inhibition of the receiving neuron. Chemical synapses allow for greater flexibility in neural signaling and are the predominant type in the human nervous system. In contrast, electrical synapses involve direct, rapid transmission of electrical signals through gap junctions, which physically connect the cytoplasm of adjacent neurons. These synapses enable synchronized activity among groups of neurons and are commonly found in cardiac and smooth muscle tissue. The electrical synapses provide fast communication, but they lack the adaptability and modulation seen in chemical synapses. Thus, chemical synapses play a more significant role in the complex neural processing, learning, and memory. The synaptic neuron that sends the signal in synaptic communication. It contains synaptic vesicles filled with neurotransmitters, which are chemical messengers responsible for transmitting signals to the next neuron. When the action potential reaches the axon terminal of the presynaptic neuron, these vesicles release neurotransmitters into the synaptic cleft, a small gap between the presynaptic, and postsynaptic neurons. The synaptic cleft serves as a space through which neurotransmitters diffuse before binding to specific receptors on the postsynaptic neuron. The postsynaptic neuron contains these specialized receptors which detect and respond to neurotransmitters, either generating an excitatory or inhibitory response. This interaction determines whether postsynaptic neuron will continue transmitting the signal, playing a crucial role in neural communication and overall nervous system function. Neurotransmitter release is a crucial step in synaptic communication, allowing signals to be transmitted between neurons. The process begins when an action potential arrives in the axon terminal of the presynaptic neuron. The electrical signal triggers the opening of voltage-gated calcium channels, allowing calcium 2 ions to enter the neuron. The influx of calcium ions causes neurotransmitter-filled vesicles to move toward and fuse with the presynaptic membrane, a process known as vesicle docking. Once fused, the vesicles undergo exocytosis, releasing the neurotransmitters into the synaptic cleft, the small gap between neurons. These neurotransmitters then diffuse across the cleft and bind to specific receptors on the postsynaptic neuron, initiating either an excitatory or inhibitory response. This sequence of events ensures the precise transmission of neural signals, enabling the nervous system to regulate movement, cognition, and various physiological functions. Neural communication at the synapse can result in either an excitatory postsynaptic potential, EPSP, or an inhibitory postsynaptic potential, IPSP, depending on the type of neurotransmitter release. In EPSP, neurotransmitters such as glutamate bind to receptors on the postsynaptic neuron, causing depolarization by allowing positively charged ions like sodium to enter the cell. This makes the membrane potential more positive and increases the likelihood of an action potential firing. promoting neural signal. In contrast, the IPSP occurs in inhibitory neurotransmitters like GABA the, that bind to the receptors, leading to hyperpolarization. This happens as negatively charged chloride ions enter the neuron or potassium ions exit, making the inside of the cell more negative and decreasing the likelihood of an action potential. The overall activity of a neuron depends on the summation, which refers to how multiple EPSPs and IPSPs interact. If the combined excitatory input reaches the threshold potential, the neuron will fire an action potential. But if the inhibitory signals outweighs the excitatory ones, the neuron remains inactive. This delicate balance of excitatory and inhibitory inputs ensures precise control of neural activity, allowing for complex processes like movement, cognition, and reflexes. Synaptic plasticity refers to the ability of synaptic connections to strengthen or weaken over time, playing a crucial role in learning and memory. Through repeated use, synapses can become more efficient at transmitting signals, a process known as long-term potentiation, or LTP, which enhances learning. Conversely, synapses that are rarely used may weaken, a process called long-term depression, or LTD. In addition to plasticity, neural communication is influenced by diverging and converging pathways. In divergence, a single neuron sends signals to multiple neurons, allowing for widespread communication and coordinated responses, such as in motor control. In convergence, multiple neurons send signals to a single neuron, enabling the integration of information from different sources, as seen in sensory processing. To maintain precise neural signaling, the nervous system also regulates neurotransmitter recycling and degradation. After neurotransmitters bind to the receptors, they must be removed from the synaptic cleft to prevent prolonged effects. This occurs through the reuptake into the presynaptic neuron, enzymatic breakdown, or diffusion away from the synapse. These mechanisms ensure that neural communication remains efficient and controlled, allowing for proper brain function and adaptability. Neurotransmitter imbalances play a significant role in various neurological and psychiatric disorders, affecting brain dysfunction and behavior. Parkinson's disease is caused by a deficiency of dopamine, a neurotransmitter essential for motor control. The loss of dopamine-producing neurons in the brain leads to symptoms such as tremors, rigidity, and difficulty with movement. In contrast, depression is also linked to serotonin imbalance, where low levels of serotonin contribute to mood disturbances, feelings of sadness, and reduced motivation. Another example is epilepsy, a condition associated with GABA dysfunction. Since GABA is the brain's primary inhibitory transmitter, reduced GABA activity can lead to excessive neural firing, resulting in seizures. Pharmacological treatments target these neurotransmitter imbalances to restore proper neural function. For example, selective serotonin reuptake inhibitors, better known as SSRIs, commonly used to treat depression, increase the amount of serotonin in the synapse by preventing its reuptake into the presynaptic neuron, thereby enhancing mood stability. Similarly, drugs like benzodiazepines enhance the GABA inhibition, increasing GABA's calming effect on the nervous system, which is beneficial in treating anxiety and seizure disorders. By modulating neurotransmitter levels, these medications help manage symptoms and improve overall neurological function. Synapses play a crucial role in neural communication by transmitting signals between neurons, allowing the nervous system to coordinate functions such as movement, sensation, cognition, and emotion. Through the release of binding of neurotransmitters, synapses enable the activation or inhibition of postsynaptic neurons, shaping how information is processed in the brain. This function is essential for learning, for memory, and adaptive responses to the environment. The activity of synapses regulates neural circuits, which are networks of interconnected neurons responsible for specific behaviors and physiological processes. Excitatory and inhibitory synaptic inputs determine whether a neuron will fire an action potential, influencing everything from reflexes to complex decision making. When synaptic transmission is balanced, neural circuits function efficiently, supporting normal behavior and cognitive processes. However, disruptions in synaptic activity, such as neurotransmitter imbalances, can contribute to neurological disorders like Parkinson's disease, depression, and epilepsy. Therefore, synaptic function is not only fundamental to the basic nervous system operation, but also critical for maintaining overall brain health and behavior. Thank you for watching, and if you would like to see any further information, please check out these resources.